Okay, good afternoon, everybody. So this is the latest of materials of a machine learning for science seminar, and it is a big pleasure to introduce you to Noa Marom from Carnegie Mellon University in Pittsburgh in the United States. So for those of you who don't know Noa, she's a graduate from Technion in Haifa in Israel. And then she did her PhD in the Weizmann Institute to then move to the United States. And she took up first a position in Tulane in Louisiana. And then actually now she moved to Carnegie Mellon where she has been there in the materials science and engineering department for about 4 years or so. And a lot of activity of Noa is really related to working with molecular crystals and molecular superstructures. And in particular, recently, she has developed a very nice program on predicting molecular crystal, which is a particularly tough problem. So, as you know, I usually ask you to pose questions during the talk. And in particular, here, the talk is essentially divided in two parts. So I really encourage you to pose the question as Noa is talking, so we can break out of the first part and have a first question session before we resume. Okay, I guess without any further ado, I just would like to give you the stage. Please, thanks a lot to be with us.
Okay, thanks for the introduction and for the invitation. And hi, everyone. So what is a molecular crystal? A molecular crystal, as the name implies, is a crystal that is made of molecules. For example, this one is aspirin. So molecular crystals are used in various industries, mainly in the pharmaceutical industry, because usually drugs are marketed as crystals of the pharmaceutically active ingredient. But molecular crystals are also used in organic electronics, in explosives, in dyes and pigments, and various other applications. So if you're used to dealing with inorganic materials, one important difference is that here, the molecules are not bound to each other by chemical bonds. Rather, they are held together by weak van der Waals interactions. And the weak nature of these interactions generates potential energy landscapes that have lots of local minima that can be very close in energy. And this is the reason why molecular crystals often exhibit polymorphism, which means that the same molecule can crystallize in different crystal structures. And these polymorphic structures of the same molecule can have different physical and chemical properties. For example, if it's a pharmaceutical crystal, a different polymorph might have different solubility and therefore different bioavailability of the drug. If it's a material for nonlinear optics, for example, different polymorphs can have different optical properties. And if it's an organic semiconductor, different polymorphs can have different mobilities and so on. And this is why it's very important for us to be able to predict all the possible polymorphs of a given molecule and their properties. So the challenge is that given a 2D stick diagram of a molecule, we now have to predict all of its possible crystal structures. And molecular crystal structure prediction is a very difficult task. And to understand why it's so challenging, first of all, the weak van der Waals interactions and the small, very small energy differences mean that we have to use very accurate methods to evaluate the relative energies of these different polymorphs. and we need fully quantum mechanical accuracy. And then to make things worse, the space we have to search is very high dimensional. So if you just look at this example here, and you try to count the number of degrees of freedom that we have in a molecular crystal, so we have just 3 lattice parameters and 3 angles that determine the unit cell. And then we have the center of mass position of each molecule in the cell. So 3 coordinates for each molecule. And then we have the orientation of the molecules in the cell. One big difference between molecular crystals and inorganic crystals is in inorganic materials, usually you think about their crystal structures in terms of packing of spheres. But as you can see here, these molecules are obviously not spherical. And so their orientation adds additional degrees of freedom, 3 Euler angles for each molecule. And then finally, molecules can have rotatable bonds. They can have conformational flexibility. And so that just increases the dimensionality of the configuration space manyfold. So the challenge is we need to search a very high dimensional space with very high accuracy. So how do we go about predicting the structure of molecular crystals? Our approach involves 2 layers of code. There's the bottom layer is what I call the electronic structure layer. And this is the layer that, given the structure and composition of your material, it will output the properties, the energy. It can do local optimization. And then you have the structure prediction layer, that's the top layer. This is the layer that searches the configuration space and decides what material to sample next. So it will generate structures and compositions and feed them into your electronic structure layer. And the electronic structure layer will output the energies and electronic properties back to the structure prediction layer, which will decide based on this information where to sample next. So this is the general approach, and it's used very widely in all sorts of computational materials discovery efforts. So now all you have to do is put your favorite codes and algorithms in these 2 boxes. So for the electronic structure, we use a DFT code called FHI AIMS. And for the structure prediction, I'm going to talk about 2 codes that we developed in my group. The first one is the random structure generator, Genarris. And the second is the genetic algorithm, GAtor. So our workflow starts by generating random structures of molecular crystals. And Genarris can generate curated sets of structures, and they're randomly generated, but with physical constraints. And so this is useful for generating initial populations for crystal structure prediction algorithms, but you can also use it for other applications, like if you want to generate a data set, a training set of structures for machine learning, for example. So the workflow of Genarris starts by just estimating the molecular volume and the unit cell volume based on the requested number of molecules per cell. And then it will identify all the space groups that are compatible with the number of molecules per cell and with the point group symmetry of the molecule. And then it will go through the space groups one by one and attempt to generate structures. So once the structure is generated, the structure goes through structure checks to make sure that no two molecules are unphysically close to each other. And once enough structures are accepted into the pool, we go through a process of sequential steps of clustering and selection to down select what we call the raw pool into our curated pool. So the first step of Genarris is to try to estimate the unit cell volume based on the single molecule structure. And to get a lower bound on the accuracy of a model that estimates the molecular crystal volume based on the single molecule, we collected a set of about 2,000 pairs of polymorphs from the Cambridge Structural Database. And here you can see a histogram of the percent volume difference between pairs of polymorphs of the same molecule. And you can see that the standard deviation of this histogram is about 3%. So basically, you can't predict the volume of a molecular crystal with a higher accuracy than the volume differences between polymorphs that are found in nature. So this is kind of the accuracy that we're trying to hit. So the most naive approach that you can take to trying to estimate the molecular volume is to put van der Waals spheres on top of all the atoms in your molecules, and then estimate the volume occupied by the van der Waals sphere. We do that using a Monte Carlo method, where we randomly sample points, and then for each point, we check if it's in the sphere or not. And that gives you the volume occupied by the atoms in the molecule. And then you can do a linear fit on top of that, as you can see here. And that gives you errors of about 4%. So to improve on that a little bit, we want to consider not just the volume occupied by the atoms in the molecule, but also the chemical composition of the molecule and what chemical groups might be participating in intermolecular interactions. So to improve the model, we added features, which we call topological molecular fragments. And here you can see two examples of benzene and glycine and how they are represented in terms of the topological fragments that comprise them. So you can see that benzene has 6 fragments that are a carbon atom bound to hydrogen. And then you also have 6 fragments that are a carbon atom bound to 2 carbons and a hydrogen. And then similarly, you can represent glycine based on the molecular fragments that comprise it. So when you add these features, so we take the whole data set and we do an analysis of which fragments appear in the data set at least 30 times, and that gives us the fragments to use for representing all the molecular crystals in the data set. And then the linear model coefficients are found by Bayesian regression. Unsurprisingly, perhaps, we find that the top fragments are HC, HO, OC, and ON. And of course, hydrogen bonded to carbon is very common, but then the OH group and the OC group and the ON group are groups that participate in strong hydrogen bonds. So, so that means that they typically play a role in intermolecular interactions that are significant, and this is why they end up being significant fragments. So here you can see the histogram from the Monte Carlo volume estimation that has 4.72% error, and that's compared to the histogram from the Monte Carlo plus molecular fragments that has about 3% error, which is pretty much the lower bound that we were trying to hit. So after we estimate the volume, we can start generating unit cells in the target volume range, which is some distribution around the predicted volume. So what we do is generate a random unit cell and then pick a space group and start placing molecules in the unit cell. So if the molecule doesn't have any particular symmetries, then you can only put it on general Wyckoff positions. But if the molecule has symmetries, for example, benzene is a very symmetric molecule, then you can also place your molecule on special Wyckoff positions, which are sites with specific symmetries. So you can see here, for example, that for benzene, because it has so many symmetries, there are many more space groups available with special positions than in general positions. So if you're placing your molecule on a general position, you can just randomly orient it, place it somewhere in the unit cell, and then use the space group symmetry operations to generate all the other molecules in the unit cell. If you're generating a molecular crystal in a space group with special positions, you have to do an extra step of aligning your molecule with the site symmetry. For example, if the molecule has an inversion symmetry, then the inversion center of the molecule has to overlap with the inversion center of the site, because otherwise it wouldn't work. So after we generate structures, we have to check if the structures are physical. And that means that no two molecules can be too close to each other. So the way we check this is by setting a cutoff distance, which is determined by a certain fraction of the sum of van der Waals radii of two atoms that belong to different molecules. And that fraction is set to 0.85 by default. So no two atoms that belong to different molecules can be closer to each other than that. So here you can see interatomic distance distributions for certain types of interactions. And the purple line here is the sum of van der Waals radii. The red line is the 0.85 of the sum of the radii. And for the very strong hydrogen bonds that you can see here, the OHO, NHO, NHN, and OHN, you can see that if you place the cutoff here at 0.85, then you will completely miss the second peak of the histogram, which corresponds to the strong hydrogen bonds. And so for these strong hydrogen bonds, we have special settings represented by the orange lines. that are meant to include the strongest hydrogen bonds in the allowed intermolecular distances. So once we've reached the target number of structures that got accepted and passed the structure checks, we have what we call the raw pool. The raw pool can contain several thousand structures, 5,000, 10,000 structures. And we want to start down selecting the raw pool. So in Genarris, the user can implement user-defined workflows of down selection from the raw pool. And these workflows consist of different sequences of clustering and selection steps. So the workflow that I'm going to show here is a workflow that we designed for our purposes, which we call the robust workflow. But if you use Genarris, you can design your own workflows and just put your sequence of steps into a procedure list for Genarris, and Genarris will seamlessly execute your procedure list. So here you can see the raw pool that's generated for benzene with 4 molecules per cell. The green line is the experimental volume, and the orange line is our prediction from our machine learning model. And you can also see the space group distribution. So the first step that we do is we perform clustering based on structural similarity using a machine learning algorithm called affinity propagation. And then we generate a number of clusters that's equal to 10% of the initial raw pool population, and we select the exemplar representative structure of each cluster in order to reduce the size of the population, but preserve the diversity of packing motifs. So we do that. We call that diversity-based selection, and you can see the number of structures was reduced to 10% of the original population, but the volume histogram and the space group distribution are largely preserved. So the next thing we do is we take the remaining structures and we do single point energy evaluation with dispersion corrected DFT. And then we perform affinity propagation clustering again, but this time we select the lowest energy structure out of each cluster. And this aims to still preserve the diversity, but also consider the fact that we are interested in low energy structures. So we do that, and then for the structures that survive, we do full unit cell relaxation, again with dispersion inclusive DFT. And then this is the final pool you get. So you can see that relaxation causes a lot of changes in the volume and the lattice parameter distributions and the final energies of the structures in your pool. And in this case, we see that the experimental structure marked by the green X is found in the final pool. So, so the next thing we do is we take this pool that was generated by Genarris, and we use that as the initial population for GAtor. So GAtor is a genetic algorithm, and genetic algorithms are a global optimization method that works by mimicking an evolutionary process. So you start by generating some initial population of structures, and then you assign fitness values to the structures in your population. And then the fitter structures have a higher probability of being selected to mate and pass on their genes. So we select the fitter structures, and then we use crossover and mutation operators to combine the structural genes of the parent structures and generate new offspring. And the idea is that by doing that over and over again, you keep propagating the structural genes that result in a high fitness. So you keep propagating the good genes until the global optimum is found. So this step, generating the initial population, is performed by Genarris. And then the genetic algorithm is GAtor. So as you may know, several other genetic algorithms exist for structure prediction of inorganic and organic materials. So I'll tell you a little bit about what makes GAtor different. One special feature of GAtor is that it has a massive parallelization scheme. And the idea behind it is that we can eliminate the traditional notion of genetic algorithm generations, which is kind of works in serial, and we can spawn several genetic algorithm replicas that work completely independently. And the only way they interact is through a shared population of structures. So each replica independently can pick parents from the shared pool, make the parents, do the local optimization of the child structure, and if it's successful, put it back in the shared pool. So we can generate many replicas that work in parallel, and we tested this up to about 250,000 CPUs at Oregon National Lab. The second special feature of GAtor are the breeding operators, the crossover and mutation operators that are implemented in GAtor. So even though the idea of a genetic algorithm is the same idea, there can be significant differences between different genetic algorithm implementations in how, in the selection of genes and in how structures are being mated. So in our case, GAtor has crossover and mutation operators that are specifically designed for molecular crystals. Some other genetic algorithms, especially genetic algorithms for inorganic materials and clusters, often use a crossover operator called cut and splice, which means you cut the two parent structures in half and then combine the two parts of the parent structures to form a new structure. That doesn't really work so well for molecular crystals because it leads to it can lead to varying physical structures. And our goal is because the local optimization and energy evaluations with DFT are very computationally expensive, we want our crossover and mutation operators to generate structures that are kind of reasonable to begin with, because we don't want to waste a lot of computer time on trying to relax structures that are just totally ridiculous. So our genes are the unit cell parameters, the lattice parameters and angles, and then we have the center of mass positions of the molecules in the cell, and the orientations of the molecules in the cell. So what we call standard crossover will just take two parent structures and mate all of these genes with random fractions. And then we also have a crossover scheme called symmetric crossover that takes the space group symmetry as a gene, and the child will inherit the space group symmetry of one of its parents. And in addition to crossover, we also have mutation operators. Mutations operate on selected genes of one parent. So we can do all kinds of things like rotating molecules, translating molecules, applying strain to the unit cell, swapping molecules, and then we also have mutations that involve permutation combined with reflections and rotations. And the idea of these mutations is that they will break or preserve some of the space group symmetries. The third special feature of GAtor is evolutionary niching. So usually when we're looking for the global minimum structure, the most stable structure, we use an energy-based fitness function such that the structures with the lower energies in your population will have a higher fitness. And this is how our energy-based fitness function is defined for the structure I. So the idea of evolutionary niching is that you can dynamically evolve several subpopulations instead of just focusing on one basin. So in GAtor, we do that by using the affinity propagation clustering algorithm to dynamically cluster the population of structures on the fly. So every time you add new structures, the whole population gets reclustered. And then the fitness of each of each structure is divided by the number of members in its cluster. So that means that the fitness of the more populous clusters gets penalized. And then you have a shared cluster-based fitness for your pool. And by doing that, by penalizing the most heavily sampled regions of the configuration space, you can steer the genetic algorithm into regions that are promising in terms of their energy, but undersampled. And you can get it unstuck from local minima. So let's see an example for how this works. So when you do evolutionary niching, there's also a question of the descriptor you use to represent your structures to the clustering algorithm, which in principle corresponds to how structural similarity is being evaluated. So one descriptor we use is called the relative coordinate descriptor, or RCD, and that captures the packing motif similarity through the relative positions and orientations of environments, neighborhoods of molecules. So the way we construct it is you pick a reference molecule, and the N molecules whose center of mass is closest to your reference molecule. In our case, we use 16 for N. And then you build this long vector of relative center of mass positions and relative orientations of your neighborhood of 16 molecules compared to your reference molecule. And that neighborhood represents your molecular crystal. Another descriptor we use is a lattice parameter-based descriptor. Actually, that was the first one we came up with historically, and it's just based on the 3 lattice parameters normalized by the unit cell volume. So that's a kind of primitive descriptor. And then a third descriptor we used is a radial distribution function for pairs of atoms. So for this target, it has a few halogens. So halogen bonds are going to be the important intermolecular interactions. And this is why we look at RDFs for pairs involved in halogen bonds. So this is the initial pool that was generated by Genarris. And this is what happens when you evolve your population with a typical energy-based fitness function. So you see here that this region of the configuration space is very heavily sampled. And this region corresponds to layered structures with these layered packing motifs. And in addition, you can see that regions of the configuration space that were not represented in the initial pool are not sampled. So when we do evolutionary niching, here it's based on the lattice parameter descriptor. And you can see that this region is sampled more frequently. And this is the region that corresponds to herringbone structures, for example. So when you use evolutionary niching, you start seeing new structural motifs. And we also get some sampling in this region that wasn't really represented in the initial pool, which it's characterized by large C parameters. And that region actually turns out not to be useful. So finally, we evolved with evolutionary niching using the RDF and RCD descriptors. And now you can see that, again, we sample this region more frequently. This region with the large C parameters is not sampled, so that's good. And with both of these descriptors, we find the experimental structure. And if you look at the structure, you can appreciate why it's so hard to find, because it has this very unique and unusual zigzag packing motif that apparently corresponds to a narrow funnel in the configuration space, whereas these layered structures apparently correspond to a large basin. So you can see that evolutionary niching can steer your genetic algorithm away from the regions represented in the initial pool and into more promising regions that are undersampled. So that can help you overcome initial pool biases, and it can also help you overcome selection biases. Okay, so our recommended best practice with GAtor is, instead of running it once for a very long time, we recommend running it several times with different settings, because you don't know a priori what settings are going to work best for your molecule. And by settings, I mean the frequency of crossover compared to mutation, which crossover scheme you're using, and which selection scheme you're using, and so on. So here you can see how the experimental structure of target 22 from the last blind test was generated through different evolutionary routes in different GAtor runs, starting from the same initial pool. So finally, after we run GAtor several times with different settings, we collect all the structures we find and remove duplicates. And then we go through hierarchical re-ranking with increasingly accurate energy methods. So here you can see, you can appreciate how small the energy differences are. For target 22, you have 100 structures in an energy window of less than 0.1 eV. And you can see that as we go, we switch from the pairwise Yurchenko-Sheffler dispersion method to the many-body dispersion method. And then finally, we switch from the PBE semi-local functional to the PBE hybrid functional, which is more accurate. And you can see that the ranking of structures changes depending on the energy method used. And only the most accurate method, which is the PB hybrid with many-body dispersion, ranks the experimental structure as the most stable structure. This is the end of the first part of the talk that was about crystal structure prediction. And I have a second part about electronic and excitonic properties. So we can take a break of like 5 minutes to answer a few questions about the first part of the talk before I move on to the second part.
Yeah, I mean, indeed, there are a few questions here. So maybe I'll just go in order actually as they appeared. So Rajarshi Tiwari from my own group is asking, in the GAtor, what is the typical population size? And does the genetic algorithm in this case sometimes show premature convergence, and what do you do in that case?
Yeah, so typically we start from, actually I don't have any genetic algorithm convergence plots in this talk, but you can go to the JCTC paper here and you can see them. So, so typically we start from an initial pool that can be like, let's say 50 or 100 structures. And then we run the genetic algorithm for several hundred steps, and usually it'll saturate within, let's say 50 steps or so. And you can see that it's saturating because you see that at some point you're no longer getting lower energy structures. And you also see at some point, once you've thoroughly sampled your low energy regions, that the average energy of your population will start increasing. And so that's where you stop your genetic algorithm run. And in order to prevent or work around this premature convergence, if it does happen, that's why we run GAtor several times with different settings and collect everything we find. Because different runs with different settings might go to different basins and find different things, especially if you use evolutionary niching. So you should always do at least one run with evolutionary niching.
Okay. Now the second question is from Sabine Kerbel, who's also was in my group, and she's asking if you could please explain again how your initial select of a suitable space group is done for polymorph, and how big is the risk to miss a structure?
So the selection of compatible space groups is based on the space groups that are compatible with the number of molecules per cell. So for example, if you requested 4 molecules per cell, you can only have space groups that will generate a cell with 4 molecules. So if your molecule has no symmetries, that means only space groups with general Wyckoff positions that generate 4 molecules. If your molecule has symmetries, then that opens up additional space groups that use where you would use special Wyckoff positions with site symmetries. And then the risk of not finding a structure always exists. And what you can do to try to mitigate that risk is, again, run several times with different settings. And that applies to both Genarris and GAtor. In Genarris, one very important setting is the SR setting for the intermolecular distance cutoffs. And another very important setting is your volume distribution. And in GAtor, again, you run with different settings, and so that would decrease the probability of missing a structure, but of course, there are no guarantees.
Okay, maybe I have a question myself, and then I guess maybe we can move to the second part. So
Yeah.
When you do the clustering in your Genarris part, I mean, you do the clustering and then essentially you reduce the size of individual cluster, if I understand correctly.
Yeah.
I mean, I just wonder, I mean, what do you find in these clusters? So I mean, you find structures which are very, very similar, or so I mean, is there a criterion in which you throw away stuff?
So the criterion in the first step of clustering is it's just based on diversity. So that means that structures that are structurally similar with respect to the representation you use are going to be clustered together, and then you can just pick one of them to have that, to still have that diversity. So that's the diversity-based clustering. And then the second selection step, we cluster again based on structural similarity, but now we also have energy information. So we pick the lowest energy structure out of a cluster of similar structures. And the reason for this is in Genarris, we generate random structures with no relaxation, at least, up until the final pool, we don't do any relaxations. So the similar structures in your cluster might be just different variations of the same structure that are, at different points away from your local minimum.
Yeah, okay, thanks a lot. Okay, so I don't see any more questions for this part, so maybe we can continue.
Okay.
I'll mute myself again.
So the second part is a little bit shorter, and basically once we have predicted possible polymorphs of our molecule, we also want to know what are their electronic and excitonic properties for various applications. So in this case, we chose to focus on singlet fission as the application. What is singlet fission? So typically in organic solar cells, a molecule gets photoexcited, and that produces an exciton, which is a pair of electron hole. And then that exciton is separated to produce free charge carriers. But in some materials, it's possible for that singlet exciton to split into two triplet excitons that have half the energy. And then it can be possible, if you harvest the two triplet excitons and separate them, to get two charge carriers for the price of one photon. And so that can significantly increase your solar cell efficiency, especially if it enables you to utilize high energy photons that would otherwise be lost to heat. So, however, we don't really see singlet fission solar cells around us. And the reason is that there are not enough suitable materials. The best singlet fission materials are acenes like pentacene and tetracene, and they're not sufficiently stable to actually be used in solar cell modules. So our goal is to try to find, by computer simulations, some new materials for singlet fission. And the main criterion for singlet fission to happen is the thermodynamic driving force. So the energy of your singlet exciton has to be greater than twice the energy of your triplet exciton. Otherwise, singlet fission is not going to happen. So this is going to be our primary criterion. So since we're interested in excited state properties, we can no longer use density functional theory, which is a ground state method. So DFT is great for getting ground state properties, for example, like the crystal structure. However, when you want to get excited state properties, you need to move on to excited state methods. And in this case, we use methods that come from Green's function-based many-body perturbation theory. The first one is called GW. GW is actually not an acronym. Here, G represents the one particle of Green's function, and W represents the screen-Coulomb interaction. What GW does is it accounts for the effect of electronic screening, and so it will account for the normalization of your energy levels in response to the addition or removal of an electron. So that can give you properties that are associated with charged excitation, which means that an electron is being added or removed. And that includes things like ionization potentials, electron affinities, fundamental band gaps, and so on. The third group of properties I have here are associated with neutral excitations, which means that an electron gets excited to an empty state, but it remains in the system and it interacts with the remaining hole. So in order to describe these properties, you have to account for electron-hole interactions. And that is done by solving the Bethe-Salpeter equation. And that gives us information on properties like optical absorption, the singlet and triplet energies that we want for singlet fission, and also the exciton wave functions. So here you can see the effect of crystal packing on the band structure of the polymorphs of Rubrene. Rubrene is everyone's favorite organic semiconductor because it has very high mobility, even higher than amorphous silicon. And it has 3 known polymorphs. The orthorhombic form is the most common one, and it also has a monoclinic form and a triclinic form. And you can see significant differences in their band structure. The monoclinic form has pretty flat bands compared to the other two, and also a somewhat larger band gap. And we can attribute that to how the molecules are packed in the crystal. So in the orthorhombic and triclinic forms, you can see that there's backbone overlap between neighboring molecules. And that leads to stronger electronic coupling between the frontier orbitals that are localized on the backbone. So that gives you dispersed bands. But in the monoclinic form of Rubrene, there are no cofacial interactions. So that leads to weak coupling between molecules and flat bands. In addition to band structure and band dispersion, the crystal packing also affects the excitonic properties, and the exciton wave functions. So what you can see here are exciton wave functions, and in order to represent them, because they're a function of two coordinates, the electron and the hole, we place the hole somewhere, and that's represented by the red dot. And then the yellow clouds represent the electron distribution with respect to this hole position. So people classify excitons in molecular crystals into two types of excitons. A Frankel exciton is where your electron density is concentrated around the hole position. So for example, here you can see that there's some yellow on top of the red. So this exciton has a little bit of Frankel character. And then a charge transfer exciton is where the electron density is distributed away from the hole position on neighboring molecules. Like here you can see that the hole is here and the electron is distributed on several neighboring molecules. So we can see trends for different packing motifs. For example, in pi stack structures, you can see that the electron is distributed on the neighboring molecules in the stack. And in molecular crystals with no cofacial interactions, the electron is distributed on equidistant neighbors. In herringbone structures, the electron is distributed on a layer of molecules in this plane here. And in sandwich herringbone structures, your electron is distributed on the dimeric neighbor of the molecule with the hole. So these are kind of qualitative trends, but we wanted to quantify the exciton character. And to do that, we developed the double Bader analysis method. So you may be familiar with Bader charge partitioning, which is a popular charge partitioning scheme for electron distributions that have one position variable. And we extended them to exciton distributions with two position variables by performing nested sums over electron and hole positions. So this is what it looks like. So this is the probability of finding the hole on molecule A, this is molecule A, and the electron on molecule B, this one's molecule B. So the innermost sum here is you place the hole on some atom on molecule A, and then you calculate the exciton wave function from a BSE calculation, and you do Bader analysis of the electron distribution with respect to this hole position, and you sum over the charges on all the atoms J that belong to molecule B. But then we also have to consider that the hole can be on different positions in molecule A. So the outer sum here sums over hole positions on atoms of molecule A. And how do we decide where to sample hole positions? We treat the HOMO orbital of the molecule as representative of the probability of finding the hole on different atoms of that molecule. So the example here is for quaternion, and because of the symmetry of quaternion, you only have to consider one quadrant of this molecule, and you can see here the Bader charges that correspond to the percent probability of the hole being on these different sites. So we sum over all the hole positions weighted by their probabilities. And then finally, the middle sum corresponds to whether we sample more than one position in the Bader volume of a given atom. So we do all that. And then finally, we calculate the degree of charge transfer as 1 minus the degree of Frankel character, which is the probability of finding both the electron and hole on molecule A. And the reason why we do all that is that excitons with charge transfer character are considered favorable for singlet fission because they would couple more easily to the triplet states of neighboring molecules. So that would be a secondary descriptor for us. So finally, we arrive at this two-dimensional descriptor where we have the thermodynamic driving force for singlet fission on the x-axis here. And then on the y-axis, we have the percent charge transfer character of the singlet excitant. And we put different materials on this chart, and you can see here that pentacene, which is the best known singlet fission material, is here in the top right corner. This is where you would find your best materials. Another thing you can see here is that both the thermodynamic driving force and the excitant character depend not only on the molecule's structure, but also on the crystal packing. We have two systems here with polymorphs. Perylene has 2 polymorphs, and then Rubrene has 3 polymorphs, 1, 2, 3. And you can see that the different polymorphs of the same molecule are in different places on this chart. So this means that it's not enough to consider the properties, the excitation energies of a single molecule. You also have to consider the crystal packing if you're interested in singlet fission in the solid state. And then we identified monoclinic Rubrene and quaterylene as possible candidates for singlet fission that are close to pentacene. Quaterylene specifically is a very stable molecule. And also it has a narrow optical gap, which means it could potentially capture more of the optical spectrum. And then perylene here, based on this chart, is not suitable for singlet fission, but it could be suitable for the opposite process of singlet fission, which is triplet-triplet annihilation. So this is where 2 low-energy triplet excitants combine into 1 singlet excitant. And that can be used for harvesting photons with energies below the gap of the absorber. So, so we can use this to try to find more singlet fission materials. We also evaluated a group of phenylated and pyrene-fused acenes, which you can read about in these 2 papers. And some of them, this is pentacene here, and some of these phenylated and pyrene-fused molecules are close to pentacene and tetracene, but are more chemically stable. So they could also be potentially useful singlet fission materials. And this is the end of the talk, and I'd like to thank the students that did the work I showed you, and also the funding agencies that supported it. And you can go to my group's website to download our code, GAtor, Genarris, and we also have a new code, Ogre, which I didn't talk about, but it generates molecular crystal surfaces. And it's going to come out soon in JCP, so you can read about that too.
Okay, thanks a million. So, okay, this is the little bit weird moment because there is nobody clapping, but okay. But actually, the people will clap on the chat eventually. Okay, so I guess maybe we can take a couple of questions for the second part. Oh yeah, here we go. We have people clapping now. So maybe just a couple of questions myself. I mean, I just wonder, can you then? I mean, in molecular crystal is as you said, is a lot of polymorphism, and is actually not that easy to drive one the synthesis of one polymorph of the other one. But I just wonder, can you eventually go the other way around? So know what sort of exciton you want and try to see what crystal will give you that exciton. So try to close the loop between the two parts of the talk.
Yeah, we're working on this.
Right. I mean, what would be the strategy then?
I'm not going to discuss research plans and unpublished work on a YouTube talk, but I can
All right.
answer your question in person if you
All right. Okay. No, fair enough. Okay. The other question is a little bit more technical. I mean, I guess so when you work at your descriptor for the exciton, essentially you have a point-like hole that you move around, and you actually look at, and essentially you move it across how the HOMO look like, right? I was just wondering. I mean, in principle, one can think about that also the hole is delocalized over more molecules. I just wonder, how arbitrary is this decision of where to put the hole? How is the structure of the hole?
So just based on the fact that in this case, and you can always verify this by looking at which transitions are contributing to your singlet excitation in your crystal. In this case, we see that the singlet exciton for pterylene is more than 99% HOMO to LUMO transition. So that means that we can, safely assume that the hole will be located in the HOMO of the molecule, and this is the justification for doing the beta analysis on the HOMO of the molecule in order to decide where to put the hole. And of course, you can always verify. You can put the holes in different places and see how much that changes your exciton character. And in fact, this is what the error bars are on the charge transfer character. They represent the minimal and maximal charge transfer character that you get from double beta analysis if you put your hole in different positions.
All right. Okay. Yeah. Thanks a lot. Okay. So I don't see any further questions. So maybe I guess we can close here the seminar. So again, thanks a million for being with us today, and thanks all of you out there for listening. And I guess I'll see you next week. Thanks a lot.
Okay. Bye, everyone.